Hi there. I'd like to get started if everyone could come in and finish their coffee and sit down. Could you bring folks in from outside too? That'd be great. Thanks. Okay. So we're going to start the second portion of the morning's talks, and I will also we'll be making sure to keep, everyone did a great job of being on time, thank you, but just to ensure that we have the same continuity, we will be handing up signs at 5 minutes and 1 minute too. Anyway, it's my privilege and pleasure to introduce David Kratville of CDFA, and he'll tell us about the Hydrilla Eradication Program. David, thank you.
Thank you. Hey, thank you for having me here. It's nice to be down out of the smoke. I actually live in Sonora, so we got rain last night, which everyone was very happy about, but driving past the Butte fire this morning, I saw about 200 fire trucks from all over the state. It's quite an amazing scene up there, but at any rate, I'm here to talk about the Hydrilla Eradication Program. And I apologize for those of you that have heard me say this before. This is my little soapbox I get on. We currently are working still work on the Hydrilla Eradication Program, but historically CDFA had numerous noxious weed programs. Where's that little thingy? There it is. As you see up there, this map on the left, I formerly was I am the last person to ever be hired as one of the district weed biologists. We used to have a terrestrial weed program as well as a biological control program statewide. So my counterparts and I, this was my district here. I had 13 counties. This is why I live in Sonora. But at that time, we were responsible for doing all the survey of both terrestrial and aquatic weeds in our districts, as well as working with county ag commissioners' offices, releasing biocontrol agents, and doing eradication work. And that's a big distinction with the Department of Food and Agriculture. Our emphasis is on eradication. Because of that, I'm sure most of you work in the control realm, where you're trying to mitigate things at a economically acceptable level, or you have some other parameter. The world that we work in eradication is we have zero tolerance. So it's hard for us to do research because of that. Particularly with hydrilla, I looked for hydrilla for 10 years without ever seeing a plant in the state because we just don't allow it to grow. And anybody in here besides John Madsen who's ever seen hydrilla? Oh, there's a few of you, okay. From out of state, where you've seen it most likely? Okay. So in addition to that, well, I should say that program, the terrestrial weed program as well as the biocontrol programs, that was all defunded in 2010. They ended in June of 2011. Which is when I transitioned over to the Hydrilla program full-time. I am actually the statewide supervisor of the Hydrilla program. But prior to me being in the noxious weed program, I was also the statewide coordinator for the purple loosestrife control program. Went from the Oregon border to the Mexican border and everywhere in between. Purple loosestrife is a riparian plant. It's found pretty much from the Kern River to down in the south, all the way up to the Fall River area around Bernie in the northeast. So it's pretty common in the state. And that was a CalFed funded program that also we no longer do. And then on sponge plant, which is this little guy right here, I believe myself and Robin Breckenridge, my old boss, were probably the first people to ever find sponge plant in the state. We controlled it, we eradicated it from a small pond in Sonoma County. And then also Madsen's second to last photo there of water lettuce. Robin and I also were responsible for finding water lettuce in California. It was actually in the Stone Lakes water treatment plant right over in Elk Grove. We removed it and then a real cold winter set in and killed it off. But that's the type of work Food and Ag used to do much more of. We used to do statewide surveys. We used to do, all my counterparts and I, we would come down 2 weeks out of the year, once in the fall, once in the spring, and survey every inch of the delta for hydrilla primarily. We have never found hydrilla in the delta, but we would come down and we sort of mapped the insurgence of kabamba and some of these other weeds that are now fairly common now. So why do we still work on hydrilla? Primarily because of this. We are mandated by the Food and Ag Code. The Director of Food and Ag shall conduct surveys and, where feasible, eradicate hydrilla anywhere it occurs in the state. That mandate, in addition to, we had a separate pool of funding. In 2011, we were funded through Harbors and Watercraft. We get funding from Department of Water Resources and also the U.S. Bureau of Reclamation. That allowed us to maintain this weed program. So what is hydrilla? It can easily fill the water column up to depths of 20 feet. When it does fill the water column, and I'll have some good examples of this plant later on, but we get this 85% reduction number down from Imperial County, which was the first place it was ever really discovered in California, where it became a real, that's what the program, when the program was funded, was down in Imperial Irrigation District. They were having 85% reductions in the storage capacity of their reservoirs and 85% reductions in their ability to flow water through irrigation ditches. So a way to think of that is if you had a 100-foot acre reservoir infested with hydrilla, you essentially would have 85 acre feet of hydrilla and 15 acre feet of water. That's the significance of hydrilla. And in a state like ours where the entire state runs on moving water from one end to the other, this is a massive risk. So this is a 30-acre pond in Madera County that we have since eradicated, but that is what a typical hydrilla infestation looks like. It is just absolutely 100% solid, full of plant mass. You can see like where ducks maybe have swam through the surface there. One of the things that hydrilla tends to do that some of the other more common invasive weeds, aquatic weeds in California don't do, hydrilla will hit the surface and then branch laterally, so it just completely fills the water column. There are times in the delta at high tide you can zip right across the agarria beds. You know, they're down there a foot or two under the water at high tide. I doubt that would happen with hydrilla. It grows so long, it doesn't limit itself when it hits the surface. I think it would just like, you know, a dog's hair standing up, it would just ride that raising tide and be a massive problem. Here's another example, and I'll show you a picture of this pond next that's empty. It just looks like a golf course, like a putting green when we were there. That is 100% solid hydrilla. And there's the same pond. We used herbicides in this pond for many years to the point that we were actually doing damage to this gentleman's trees. So he agreed to let us drain the pond, and we removed a bunch of the sediment and lined it with a pond liner. But you can see the depth of that pond. Down here where the spot is, the pointer, I would say it was easily 15 to 20 feet deep, and it was just 100% solid from the bottom to the surface with hydrilla. So this is hydrilla here on the left. This is the real distinguishing characteristic that we always point out to people. Whereas Elodea, real common, native, there's 2 species native in California, will normally have 3 leaves in a whorl. Agarria is a weird one. It can have anywhere from 4 to 9. But hydrilla almost religiously has 5 in a whorl. One of the things is you can look at different pieces of the plant, and depending on how far the nodes are, if they pack together, it can be hard to see. But when you look down lower on the stems, quite often you'll see these rings of 5 leaves. Also, hydrilla is heavily serrated along the margin of the leaf. And then there's these little, I believe they're called stipules on the midvein, these little spines on the bottom of the plant. So the big test I always give anyone is, does anyone know what any of those plants there on the right are? Does anyone know what this big one is? That's Agarria densa. That's what's all over the delta. You ask any bass fisherman in the delta, and that's hydrilla. But it's actually Agarria densa. Like I said, we've never found hydrilla in the delta. This is coontail, Ceratophyllum, which was one of the native ones that Valerie was talking about. This is another little native plant, Elodea. It tends to have the whorls of 3, and then this is our public enemy number 1, hydrilla, right there. It's actually, when you look at the leaves directly, it's a little bit smaller than Agarria, which can be surprising to some people. There's also 2 forms of hydrilla. There's both monoecious and dioecious, and they both occur in California. So it's very variable, but that's just an idea of what they look like together. So here's a little example of some of the ways that hydrilla can propagate itself. As has been said earlier, many plants have ways of spreading themselves within a water body, or in the case of hydrilla in particular, overwintering in a dry season. So the reason hydrilla is such a problem for us is these down here, the tubers. So it evolved in an area that gets monsoon rains, and when those quickly filling lakes dry, this plant had to have some way of, I call it overwintering, or that dry period. So it developed these tubers, which it sends down. When we dredge them, I've seen them in California anywhere from 18 inches to 2 feet deep. They tend to go down to where they hit a clay layer, in my experience. But that tuber can sit down in dry soil for 4 to 7 years easily. I wouldn't be surprised if they lasted longer than that. You can dry a pond with hydrilla in it, leave it dry for 3-4 years, come back, fill it with water, and boom, you got hydrilla again. The other aquatic plants in California, I'm not aware of ones that will do that. Likewise, when we go in and treat with chemicals, we can very easily kill the majority of the weeds that are actively growing. But the next spring, the thing that comes back is hydrilla. So there's no one-shot cure for hydrilla. Another way that it spreads, these are not overwintering techniques, but these are called turions. It's actually a little compacted little bud that snaps off very easily. It breaks off, and those sort of disperse within the water column, within the water body, as well as with most all aquatic weeds, fragments. Just any, if you have 2 or 3 whorls, 2 or 3 nodes worth of plant material, you're likely going to start a new plant when that lands in some sediment somewhere. In addition, hydrilla also has what can be sometimes perennial root crowns. So you can go down and the plants break very easily, and you leave those root crowns as well as tubers in the soil, and it says thanks for the haircut and keeps growing. And then hydrilla also forms rhizomes. So short of seeds, it basically does every method it possibly can to propagate itself. So here's an example of a tuber. This was actually an old tuber that sprouted. You can see they look like a little small compacted bud, and then that little terminal tip started growing out of it. Once it hits the soil of the surface, it begins forming this root crown and then splitting. And here is this is why hydrilla is, again, so hard for us to kill, is the tubers. We don't have fumigation techniques available anymore in aquatic settings in California, so we go in with dredges where we can and manually remove the tubers. So here again, those are tubers on the right. These are newly formed tubers. So this plant shoved these down into the soil, and this is for next year's crop, or 10 years from now's crop. And then these are the little terminal turions, and these will break off easily and spread throughout the water column. They're both actually the same thing. The tubers form also at the tip of a shoot, but towards the fall or a little bit later in the year, the plant actually grows down and almost like a peanut does. It grows down into the soil and then forms that tuber down beneath the soil surface. And here's an example of that. This was there's a root crown. You have rhizomes starting a new plant, and then a rhizome to a new plant, and then these are the shoots going down and forming tubers below. So in California right now, as of 2015, we have several active eradication projects. Our largest one by far is up in Clear Lake. We have the other largest one, I would say, is up in Yuba County, near the town of Oregon House. We have a couple active ponds in Nevada County, and then up off the Sacramento River near Redding we have some. The ones near Redding are approaching eradication. We have not seen plants there in a number of years. We've actually backed off herbicide applications, and the technique we use, it's similar to a pre-emergent in a terrestrial setting. We put out these slow-release pellets that form like a layer of herbicide. So as those tubers begin to sprout, that's the first thing they hit and they die. But because of that, and how persistent that chemical is, it takes some time for us to be able to say that we've eradicated it. We don't know if there are tubers sprouting and being killed. So we quit making applications after 4 to 5 years of not seeing any plants, and then we go into a survey mode, waiting to see if any plants come back. And sometimes they do in that 3rd, 4th, so maybe 7, 8 years after the last plant we've seen, we might have a tuber sprout, and then we go right back into this 10-year cycle if any tubers are formed. So just here's a little history of hydrilla in the state. As I said in the beginning, Imperial County, there's 600 miles of canals and drains that were infested, and I will give you an example of that. Lake County, Clear Lake, that was found in 1994, Nevada in 2004, up in Shasta County. Those are the ponds that are active, and then Yuba County as well. So hydrilla will grow anywhere in the state of California. We found it down in San Diego County, we found it in Shasta County, we found it on the coast, and then up into the foothills. As I said earlier, we used to do quite a bit more surveys. We used to attempt, the noxious weed program staff would attempt to survey every single mile of the delta for hydrilla, and we used to monitor any weeds we saw, and actually the next couple of speakers are going to talk a little bit about the mapping efforts. We've been able to give some data, some historical data anyways, on where we used to see plants. And then we've also contracted with DWR. These were some surveys that we did. We're hoping to do some of this with boating and waterways as well if we can. While our crews are out there looking for hydrilla, they're just marking down all the other plants they come across. So this is, I was asked to try to tie my talk into research for you folks. This is one of the areas that we are very concerned about is the salinity tolerance for hydrilla. I've heard it can exist up to about 5 to 7 parts per thousand, whereas Aegeria is limited from 2 to 5. So what we're getting at there is that there's a point around Antioch, Rio Vista, where you don't really see Aegeria densa anymore in the delta. I have concern that hydrilla could grow a ways past that. Hydrilla could also very likely grow at a much greater depth than Aegeria does in the delta. So the parts of the delta that we think could be susceptible to hydrilla are much more, much vaster than where Aegeria can exist. This was some work that we found. This was a modeling assessment. They looked at, I believe this was dioecious hydrilla, as it's from northern eastern U.S. But they looked at the suitability of the United States, various different models based on the native range of the plant they were looking at. This is all hydrilla, but whether it was from Korea, Japan, Australia. The thing to note is that California is bright red in every single one of these models. So as we've seen, California is very suitable for both growing the northern variety of hydrilla. As you can see, there's a gentleman from Florida who can tell you this more than me, but Florida is very suitable for growing hydrilla, just not this variety. It's blue there. So California can grow hydrilla, that's the bottom line. Something else we've begun doing is taking baseline DNA samples. We have concern to see if we do get a new hydrilla infestation, was it moved within the state? Was it perhaps from a different state, particularly Florida? They have resistance to our primary herbicide, fluoridone. We would be very concerned if it showed up from Florida for that fact. But these are the samples we've taken so far. Clear Lake, we've sampled 10 sites of monoecious. Oregon House, we have 2 active sites we were able to sample plants from. And then again, up in Redding, we haven't seen hydrilla in a number of years. And then in Nevada County, we have the dioecious form that we've sampled. So we're building a baseline of what our DNA looks like of our current populations, and hopefully we can use that going forward to see where the plants are coming from. So as I said, the tubers are the biggest problem for us in eradicating hydrilla. So this is one of the techniques that we use. This is a modified gold dredge. This is up in Yuba County, up in the foothills near that Oregon House. You can imagine driving through the foothills with a gold dredge and you're in state vehicle. No one thinks you say you're looking for weeds and they automatically think you're lying, but this is what we're doing. This gentleman up here, he has the nozzle. He's down here in this ditch sucking out the sediment. And then we have these screens, and this gentleman is picking any plant fragments or tubers out and then disposing of them. There's just another shot. So as you can imagine, we used to hike these things all over the place into very rural areas to suck the tubers out of the soil. So if you can remove that tuber from the soil, you're saving yourself, 5, 10 years. Ooh, I'm running out of time. We use screens as well as numerous chemical control methods. We use every type of watercraft you can imagine, as well as hiking up and down. That's actually me spraying purple loosestrife years ago. That's not Hyacinth, obviously, or Hydrilla, obviously. But the chemical methods we utilize contact herbicides for a burn down. Whenever we find an actively growing plant, we use something like copper or perhaps endothol to quickly burn that biomass down and reduce its risk of spreading. Also, this is an irrigation pond on a vineyard, so we can't use some of our systemic herbicides If that water is then going to go be put on a crop, that really limits you. So this is an area where we use contact herbicides. And as I said before, the systemic, the fluoridone pellets, we use a slow release pellet. We apply them once every 4 to 6 weeks. And we slowly maintain that carpet of pesticide right at that, that soil surface. So any sprouting tubers will hit that and die, hopefully. As I said before, fumigation was our best tool, but that is no longer legal for use in aquatic settings in California. We've tried this a number of times. This is actually gunite or shotcrete. It's like what the pool liners are made out of. So this was that irrigation ditch where the gentlemen were using the gold dredges. We had a company come in and remove soil and contour it and then spray it on just like they would do a pool. We've also used pond liners in those situations. Again, if we can seal off the tubers from the water, eventually they should just wear out. We have had these liners rip 5 years down the road, and boom, there's a big wad of hydrilla comes up through it. In Imperial County, and they get cracks in their cement liners, and hydrilla comes up. So it's down there, and it's going to be down there for many, many years. But as I said, this was Eastman Lake. This was an Army Corps lake, I believe, that they drained and fumigated. This is the little, I believe it's called Lake Murray in Marysville. Again, we drained it and fumigated it to kill those tubers. And in a situation like this, we can declare eradication in 3 to 4 years. We've been trying to eradicate hydrilla in Clear Lake since 1994. So if we could fumigate or find a replacement for fumigation, we would have much better odds. This is an example of the Imperial Irrigation District. That's what an 85% reduction in storage and flow looks like. They just literally could not move water through there. So in that situation, we were able to introduce triploid grass carp, which feed upon the plants. The Department of Food, Fish and Wildlife is very particular about where you release a non-native large herbivorous fish like triploid grass carp. But this is an example. This is an exclusion pen. So you can see the carp are able to eat all the vegetation in this area, and this is what it looked like prior to that. Also, we do still have a biocontrol program of sorts. I believe Patrick Moran's going to talk a little bit later. We are assisting them in growing some hyacinth biocontrol agents. That is the one little aspect of biocontrol that we still have. And I'm almost done. So the last thing I want to talk about that is a concern for us is this is Clear Lake. And this is an area called the Narrows. We're hoping to do some water flow studies, some dye studies in there to see where the herbicide moves. Primarily, water comes into Clear Lake here and then flows out through Cache Creek. But year after year, this is a trouble spot for us. We just keep finding hydrilla in this area called the Narrows. And there's like a large eddy, perhaps, as it comes. The water just flows through that narrow gap. But depending on the temperature changes, the water can flow different directions. And this is very akin to what happens in the Delta with the tidal changes. That's always been a huge concern of ours. If hydrilla ever were to end up in the Delta, how would we maintain that pesticide presence in an area where the flows repeatedly shift back and forth? So in closing, the big things we need is a replacement for fumigation, whether it be some other form of steam, irradiation, something like that, some form of tarping at the bottom of a water body and a different chemical. The DNA tracing of populations so we can map introductions where they're coming from and also look for resistance. And then dealing with herbicide resistance. As I've said, we have one silver bullet fluoridone that we've used for years and years. Florida did something similar, and now they're, the #1 tool no longer works in some of their water bodies. And then, as I just said, dealing with a tidal flow or a high flow situation, which luckily we've never had to do to this point. So that's it. Thank you. Do we have time for questions or no?
Time for one question.
Anyone? All right.
Thanks.
Thank you.
Okay, thanks, Dave. Okay, our next speaker is Dave Bubenheim from NASA, who's going to talk to us about sort of tools involved with mapping water hyacinth, map and user in analyzing hyacinth presence and distribution. Thanks, Dave.
there's just a trackpad. I'll let you finish. Hi, thanks for inviting me. I am going to talk, or first I should mention, I'm Dave Bubenheim from NASA Ames down at Moffett Field. Been involved with the invasive plants and Department of Agriculture for a number of years, working in terrestrial invasives as well as aquatic. The particular work we'll talk about today is part of an USDA-supported area-wide project that Patrick Moran will probably give you a little more detail about whenever he speaks. And I am going to talk about the mapping tool for hyacinth, but really as an example. What we're really involved in is trying to show how we can adapt remote sensing techniques, move it from a research tool into an operational support tool, and make management of aquatic weeds more effective and more efficient. I also want to point out Christopher Potter, who's also at Ames. He's actually the remote sensing expert. I'm a plant physiologist, an ecophysiologist. I do enough remote sensing to get myself into serious trouble, and Chris is always there to help me out. So we work together quite a bit. So tell you what I hope to be able to do is first really give you an idea of how we're trying to transition remote sensing from R&D into management decisions support. Use the water hyacinth mapper as an example, see how it fits into how well it works, how it fits into operations and management, and how you can also be used in some analysis. And we don't want to forget about the submerged plants. Actually, when we started this project, we thought we were going to be working on submerged plants 99% of the time. And then things went kind of crazy, and hyacinth became the big target. And so we've been doing a lot more work on hyacinth, but we're not forgetting about submerged. And then try to pick up at the very end a little bit about what I think is some of the most exciting part of this. It's not the mapping, but it's understanding how the plants are responding to altered environments, and then being able to use that understanding to maybe get ahead of the situation instead of always just trying to figure out where they are and how do we go kill them. So we started off, this is a version or picture of a Landsat satellite. It's a series of satellites. Landsat's been up for 20 years, I guess. And there's been decision to continue to provide continuity in renewing satellites over time. It's been a workhorse, and it flies over this part of California about every 16 days. So at about 2-week intervals, we get a nice shot of the delta. And you know, when you go out and you look at these expanses of hyacinth, get that out of the way. When you look at those expanses of hyacinth, it's always been difficult, talking to people, to actually estimate how much is out there. You know, people go out in the boats and they go around, and most of you know the difficulty actually trying to come up with a kind of authoritative number, reference number of what's actually there. And so that was part of our effort. In the process, we realized that we could actually come up with a lot more information. But so what we did was we took some basic work that was originally done here at Davis and Susan Houston's lab. And Susan's been very good about sharing things back in the 90s that provided some base signatures for hyacinth as it separates from other plants, and we were able to use that to process the information coming from Landsat, and now we routinely, every 2 weeks, make these maps. And what this map is everywhere where you see the dark blue, that is, each one of those is a 30-meter pixel, where at least 50% of that pixel is covered with what we think is hyacinth. What we're pretty damn sure is hyacinth in most cases. So this is a big image of the delta. It's a nice kind of way to get a handle on just looking at the expanse and how blue things are. We can zoom in on certain areas and draw limits around, so it's useful. We're able then to be able to identify certain functional areas. This is more of a hydrologic unit kind of designation, but we were working with boating and waterways to adapt these also to their treatment areas. But then we can come up and tell us what the acreage is. You know, at this point in this map, this is a little under 1,000 acres of hyacinth there. And that's probably a little underestimate of total acreage because these are pixels that have at least 50% or more. So there is more hyacinth out there. Right now it is, I should say, we started last year to put this together, and it was about October when we had a really functional tool, September, October. And we started in October going out with boating and waterways to look at how well it was working. And the one thing was very clear right off the bat is when the mapper told us that there should be a big patch of hyacinth around the bend, we'd go around the bend and there was a big patch of hyacinth. It's been very reliable in doing that. And so this summer, Boating and Waterways has been working with it to try to figure out how it can fit in to help them in their system. It's also being used to help identify where and when mechanical harvesting would work best, and also helping the project determine when and where to apply or release biological control. I'll give you a quick idea of some of the validation that we did. So we'd go out in a boat and we would look for these big patches of hyacinth and try to look at what the biomass estimates are. So not only are we getting an area estimate, but we're getting how much mass is actually there per unit area. And we'd go out and look for these big patches and say, well, that's 80% coverage, that's 90% coverage. In this whole process, we got a nice correlation with that. But a big limit here was people being able to actually assess what coverage is, especially when you get over 80%. And so that's is maybe a little bit of a work in progress. We're not completely finished because we're in the midst of still continuing to quantify this validation in terms of the biomass. This is some recent data collections that we made where John Matson and a group of us went out and were hanging over the boat and collecting samples. But we'd go to a site and it was a day just about the time when the satellite went over, so we'd have the satellite prediction of what biomass would be, and then the green is what we actually sampled, and in most cases we're coming pretty close. Since I was the guy cutting, I was probably too anxious and cut a little extra hyacinth outside the square at that time. Example of how this can also be used, there's decades of archival data available from Landsat. So we can go back in time and kind of and do an analysis. There's a lot of great information in this archive that we're just beginning to touch and work with Boating and Waterways and folks to try to identify what the most important questions are to ask right now. But this is just looking at 2013, 14, 15, and the kind of cycles that we've seen. And so you can see that this last dot out here in early September is already above where it was last year, and the peak last year was more in November. So who knows where we're going to be out here. At the same time, Boating and Waterways is treating more area than they ever have before, and so maybe that could have been a lot higher. But so those are the kinds of things we're trying to be able to do with or help add to analysis with this tool. This is just kind of a time course here to think about how you could use this map. 1989 was a year without a whole lot of rain, and we can see a certain degree of hyacinth. 96 was a moderate year. 2005 there was more hyacinth. 2010 was a year when we had a lot of rain, a lot of water moving the hyacinth out. There was less that year. 2013, you can see drastic increases, and this, I put the wrong date on it. It's not 2013 again. It was 2014. But so these are the kinds of things. So those are just images looking at the same date year to year. But basically at 2-week intervals, we have all this information available that we can go down to very detailed levels. We can go down and look at the area for a marina, and you can look at the dark blue, and you can look at that, and we can follow changes. So we can do these kinds of things for marinas. Another item is looking at some of the ag barriers and the emergency barriers. We can go in and look at before, we can follow that before, leading up to it, and after, and also help to try to understand where that hyacinth goes once it leaves, and that can help prepare for mechanical harvesting and problems in places like Stockton and downstream. Just an example of how we can look at, say, Middle River, or where there are these ag barriers that are put in place. Look at the images before, look at the images after their removal. We can see that the hyacinth's gone away, and we can use the rest of that map to try to understand where those things have gone. Again, not forgetting totally about the submerged, but the submerged are a big problem, because when you look at hyacinth, you can see hyacinth, but when you look at agarria, sometimes you can see agarria. That's a much harder nut to crack. It's a much harder set of remote sensing to try to track, and there'll be some more talk about mapping these things, but being able to map them on a routine basis is difficult. It's something we're definitely working on, but there's still a considerable amount of research that needs to be done to identify both the spectra and the way of teasing out of that spectra from available observations that we can get to on a regular basis, to be able to get useful information that we can use for management. It's one thing to fly over a hyperspectral instrument once every year. It gives you an idea of what's going on and figure out how to find these things. It's another thing to be able to get information that's relevant to making management decisions on a daily or seasonal basis. So what I wanted to do is now turn a little bit to what some of the other work that we're doing in terms of the plant growth. And we have a set of some very sophisticated controlled environments that we have at Ames. We originally put them together to develop life support systems so we can control many environmental characteristics. And so it's been a wonderful tool for being able to change environmental conditions that are relevant to altered conditions in the Delta. And we grow hyacinth in these, as well as aquatic plants, and we're able to do some work with the aquatics, but we're concentrating on the hyacinths at the moment. So we end up building, identifying how the plants respond to altered environments. And we try to build in to supply the parameters to a model that are really looking primarily now at water quality, the hot items that we're concerned about in the Delta, how water quality affects the plants, and how these environmental inputs and inputs that are altered in locations in the Delta, how they affect both hyacinth, and we're using a base model for submerged plants as well, and how they impact their growth and development and how that can control growth. So we end up building these response surfaces that really define what is the possible magnitude of the effect of altered conditions. What kind of impact is possible to see in the plant? And it allows us to do kind of hypothesis-driven science to build these response surfaces rather than just observational and trying to make a lot of assumptions. Now, it's not final because then what we can do is take those response surfaces and that understanding and go out into the field, go out into the Delta, and look for the relevant situations to validate that what we're finding is indeed indicative of the Delta. So then what we try to do is also understand what's happening on the land and what those land practices have to do with inputs from the land into the water. For interest of time, I won't go into a lot of detail about how we do that, but we have a series of land use models that look at the practices and look at the runoff and from different kinds of uses that are coming in, coupled with sampling that is actually done in the Delta and the relevant criteria that we're looking at for plant growth. And our intention is to then produce not maps of where the plants are, but maps of risk factors and risk zones. And whenever we're able to start to provide those areas of risk, then it not only allows us to identify targets to come in and treat, but it should provide a little insight into what is the cause, what's happening on the land or what's happening outside that's providing some input that we can try to go back and affect. So that's very much the approach that we're trying to take. It's more of an integrated approach, and I'm just about finished. And to be able to respond both to the immediate terms of management now, but also try to provide an understanding and an analytical tool that can look at what we need to know to be a little more strategic in management of these plants in the future. So I think we'll have to save a few minutes for questions, and be happy to take some. I think the question was, we have decades. I said we had decades. How far back does it go? I think we have about 20 years easily accessible. Now, I should point out that I will mention, that goes over every 2 weeks. There are other satellites that are operated commercially or by other countries that go more often. They cost money, and they're a little more cumbersome to get to. So we usually, in this kind of effort, try to go with something that's already being provided. It's free, it's routine, it's high quality, and we're paying for it already. So we try to use this first. So there are lots of sources. I think the real difficulty now is asking the questions. What do we want to go back into that for? We could probably support 50 grad students and a whole bunch of postdocs asking a whole bunch of questions that are relevant to the Delta. So,
Dave, you mentioned as you are trying to develop your predictive risk models of where the hotspots might show up, and you use the runoff quality data. Are you using data from the Irrigated Lands Program, from the IEP, or are you generating those data yourself?
Yes. So what we do, it's a combination. We are trying to look for anybody who has data that can be brought into that model. We use as a base a USDA tool called, it was originally called the Soil Water Assessment Tool. It's a great modeling tool that was developed about 20 years ago originally for erosion and runoff. And what it's a great tool for is it allows you to define what's going on an area of land. It's got land type, slope, land use. You can lay over plant cover. You can lay climate and weather and rainfall. And it looks at erosion and water retention in the soil and movement. It's been used a lot in the Chesapeake and the Mississippi, looking at agricultural pollution. So there are some canned routines in there. We're building a number of custom routines in there that define what the particular practices are in the Delta, and we're looking for information that we can turn into GIS layers. So information is GIS friendly, related to fertilizer applications, insecticides use, any kind of pesticides, fungicides, herbicides, all those. So there's an element of that we look at the land use, and the model generates a baseline of what we would expect to come in, and then we try to check that with what monitoring is available. We would definitely like to contribute to the people who are planning to put out more sensors. We would love to be able to contribute to the selection or identification of what else you might sense, how often it might be looked at, and where. There are issues that have to do with things like diurnal cycles and some of those questions that some of the sensor networks that are out there can't really give us. So we're trying to identify what is the biological response to that that's relevant, and then maybe that would provide some direction into sensor selection and placement in the field later on. Long-winded, but okay.
One more? Okay,
Cool.
thanks. Okay, great. Well, thanks. We have one more talk between here and lunch. So, Shruti Khanna from UC Davis, yay, telling us about aquatic vegetation in the Delta, the latest findings from the 2014 flyover. Come on over. Thanks.
Should I? Hi, I'm Dr. Khanna from Dr. Houston's lab, and Dr. Houston's lab has been working in the Delta since almost 2003 with hyperspectral data, and last year, due to funding from the Department of Fish and Wildlife, we got a chance to restart some of the work that had ended since 2008. And so today's talk is going to try and give you an overview of now 11 years of data and its analysis, remote sensing data, basically. I've been involved with this project also since almost 2005. But a lot of people have worked on it and got their PhDs out of it. Okay, so there was a bit of introductory slides I had in there, which I think don't make a lot of sense now, since you all have been hearing this since morning. So I'm just going to skip over some of them. Of course, the Delta is the primary gateway for biological invasions in the western United States, and that's why we have a massive problem with invasive species. This is an example of a channel clogged with Aegeria, and this is actually several years old. This is from some of our old field data collection, so sometime between 2004 and 2008. The conditions today in terms of submerged, just from our field observations, do seem to be getting a little different. This is also, some of the stuff has been covered already, but in brief, it's native, Aegeria densa is native to Brazil and Argentina and has been in the Delta for 50, 60 years now. And it's a freshwater perennial, but we have in the past found it as far as Big Break. It's mainly light limited and cannot tolerate high turbidity. However, it is now there are a lot of studies that have showed that it by itself has a positive feedback, whereby it increases sedimentation, reduces water velocity, and is basically furthering its own growth. We have found Aegeria growing up to 13 feet deep in relatively clear channels, and by this I don't mean that the depth of the water is 13 feet. I mean that the weed is, the top of the weed mat is that deep. So it's really, it can grow quite deep, and there are other studies that have recorded it as deep as 16 feet. Why do we care? It reduces oxygen levels below the mats, and this is almost true of anything out there, even the floating mats. And it can hinder the movement of fish between subtidal open water and tidal wetlands. There are a lot of other reasons, but in short. So there are other submerged vegetation in the Delta. It's quite a diverse community. Some of them are invasive and some of them are native. So among the invasives, the one other major concern, especially this year, that we have noted its expansion in Liberty Island is Eurasian water milfoil. And then there's curly leaf pondweed, which also we are seeing a lot of. I'm actually right in the middle of our field data collection in the Delta, so I'm speaking from like the past couple of days of experience. There's also kabamba, and then there are other natives such as sago pondweed, coontail, American pondweed, and elodia. So it's quite diverse out there. And of course, we have a lot of algae. And we have it in terms of floating mats. We have it in terms of epiphytic algae, stringy algae on the SAV. SAV, by the way, and I think I'll get quickly into the habit of calling this SAV, so I might as well make it clear. It's submerged aquatic vegetation. So if I slip into saying SAV, you know what I'm talking about. Of course, past 2 years, we have heard a lot about microcystis in the Delta, and we have been noticing a ton of it out there, both last year and this year. In fact, anecdotal evidence, we've almost noticed an increase this year. Just want to make a point before moving on that it is possible to differentiate between submerged macrophytes and microcystis because their chlorophyll pigments are different. So there is, microcystis does have a concentration of phycocyanins which have absorptions in different regions of the spectra, and so you don't really confuse those two. This is an example of a channel clogged with water hyacinth. This also is a very old image, but is just as true today. The problem with remote sensing of any of this vegetation is all of these species among themselves are very diverse. So when you look at water hyacinth, for example, you look at a vegetative mat which has no flowers, you look at a mat that is flowering, you look at a mat that is being treated and is senescing, and all of these have a huge variability of spectral signatures. So it is not a trivial task to try and get at the species level while classifying these species using remote sensing. And sometimes, people have started thinking of remote sensing as the magic bullet somehow, that, it can do everything. But sometimes it's hard to remember that there is a lot of complexity in the system. It's not as easy. Water hyacinth has been around for over a century, and it has the highest growth rate of any vascular macrophyte. It's a freshwater plant and can tolerate salinity to up to 15% of seawater. It's very susceptible to frost, and also to it needs sunlight. It's highly plastic in morphology. It again depresses oxygen levels. It can literally, and this is something that Dr. Madsen talked about too, it can make the water column literally devoid of oxygen. It also is host to a different composition of invertebrate communities, which travel up the food chain, and you got a problem. Now, in this talk, I especially want to bring attention to this other floating species, which hasn't caught a lot of attention in the past couple of decades. And it is becoming this, according to us, from our last 2 years of analysis, especially the new analysis that we have been doing, is the problem of the future. And it is good if we recognize it today rather than a few years down the line, where we are in that, what was that, the exponential growth part of the growth curve. So water primrose, there's about 4 to 5 species out there. Ludwigia is the genus. It again can be vegetative or flowering. Again, the spectra can be highly variable. So it's been documented at least for the past 25 years. It was considered, it has picked up growth just in the last 5 years. And it's more recent than water hyacinth or any of the other invasives. It's also freshwater, susceptible to high salinity, grows in slow-moving or still water. And again, it's highly plastic in morphology. It creeps on land and stands erect in water. And this is something we have been noticing, especially in Liberty Island, where primrose is really on the rise, is that it will climb all over the emergent vegetation. It's literally, I get the feeling, and of course our data will show it once we're done with the analysis, prove me or disprove me, but it almost makes me think as though it's killing off the emergent and taking over, kind of doing a kind of backward succession from, almost emergent to floating back again. But we'll see if that pans out. I'm not sure. This is just my idea. Same thing, and the other thing about primrose is it allows mosquitoes to breed and is a cause of concern because of the West Nile virus. There are other floating species in the delta, more native. There's pennywort. And this, again, in the last couple of years, we have noticed a drastic reduction in pennywort, even in field data, compared to the last time we were out there, which was between 2003 and 2008. And, of course, there's plenty of azolla, duckweed out there. So there are other floating species that... and the reason we need to know this is because when you start saying that, there is this much amount of water hyacinth out there or primrose out there, you need to recognize that unless you separate these species, you're kind of getting a rough estimate. You're not getting the accurate estimate. The data that we have, so from 2004 to 2008, we had HiMap hyperspectral data, which is an Australian company, and then, and I'll go into what hyperspectral is. I have a slide on that, so I'll make it clear why this is different than Landsat, for example. And we have, last 2 years, we have been getting AVRIS next generation sensor. This is a sensor that JPL operates under the guidance of NASA. We have the spatial resolution of this data is somewhere from 2.5 meters to 3 meters, depending on which imagery you're talking about. We have almost 61 to 65 flight lines taken every year, and each of these flight lines is like 4 gigabytes in size. So we're talking about a massive amount of data. Temporarily, this data has been collected every year from 2004 to 2008, every year at the same time. And then last 2 years, it has been collected more in fall. Last year, it was November, and this year, it's being collected right now. Spectral resolution of this data is, if you're talking about average NG, it's about 5 nanometers, and if you're talking about HiMAP, it's about 10 to 15 nanometers. Number of bands, again, is over 100, goes from 100 to 400, depending on which sensor you're talking about. So what is spectroscopy, and what is a hyperspectral image? I talk about hyperspectral remote sensing. And what it is that for example, you take a picture from a digital camera. In a sense, a digital camera is also a remote sensing instrument. It's not touching the thing it's taking a picture of, but it has RGB, right? It has red, green, and blue. So those are the 3 narrow little bands that you are seeing through your digital camera whenever you take a picture. And this is what a hyperspectral image is showing you. It has 100 to 400 bands in a range, a lot of which you can't see with your naked eye. So it is giving you a lot more information than just an optical image. And this is just a way of showing it that you have, so this is Venus cut in the delta, and what you see is that there is that information that you're looking at is a true color information. So you're looking at RGB. However, there's a whole ton of information behind it for each and every pixel on that image. So why hyperspectral? Why can't we do this with simpler stuff, just get aerial imagery or something like that? So spectral resolution, you get a lot more differentiation in the spectra of these species because you're looking at more than 100 bands rather than only 7 bands as in Landsat. Although Landsat also takes the entire range, though. It goes all the way from visible to shortwave infrared. So it's encompassing the range of wavelengths that hyperspectral imagery would, but it has less bands. HiMAP has 126 bands, average NG has 432 bands. How does this fine resolution data help us? First of all, what it does is, so when you look at water hyacinth versus water primrose, your eyes can make out the difference, but your imagery cannot, because you're looking at, let us say, even if you're looking at a 3 meter by 3 meter pixel, remember that that 3 meter by 3 meter pixel looks like one solid color, right? So you cannot see the texture of the vegetation. So when you look at water hyacinth, you know that's hyacinth and you know that's primrose, but you can't make that out in the imagery. Your multispectral imagery has much wider bands, which doesn't allow you to see some of the finer features that can allow you to differentiate between these species. How is that? That by looking at hyperspectral imagery, you can look at nitrogen content, you can look at water content, you can look at pigment content. And in these matters, in the finer biophysiological characteristics of these vegetation, there are differences between water primrose, water hyacinth, pennywort, all of these species. So you can actually differentiate them at the species level if you have access to looking at this fine quality information. For example, I'm going to go through 2 examples showing the importance of spectral range and spectral resolution. So Dave talked about some of the complexity of getting SAV out of separated from everything else, right? So we have submerged aquatic vegetation, because of being in water, absorbs light completely in the shortwave infrared, and so it has very low reflectance in that area. And you have every other species, including all the way from tules, cattails, so emergent vegetation, all the way from soil, even dry vegetation, or floating, whatever you take, all of it is higher in SWR. And so you can tell the difference. This is a histogram of SAV and water versus emergent and floating vegetation, and you can see they're very different. So SAV and water have much lower reflectance in the SWR. Similarly, why do we need spectral resolution? So you have what is called in vegetation a red edge, wherein the reflectance of vegetation goes up tremendously from the visible to the near infrared. In the near infrared, it actually is reflecting a lot more light. So this is what is known as the red edge in remote sensing. And what you see is that you see it even in the submerged vegetation. So here I have 3 signatures. The green one is showing, the floating algae mats that you might remember that I'd shown a picture of. Then you have really thick mats of agaricus, and this is the red signature is what that would look like. And then you have really low, so really sparse salve vegetation, and even that has a bit of a red edge. Whereas if you look at water, at the same region of the spectra, the reflectance is actually decreasing. So if you look at a narrow band index, like the red edge index, you pick 2 bands right in that on that slope, and what you have is again that you can tell these 2 apart. Similarly, for water hyacinth, what you have is water hyacinth is a succulent. So it has very thick leaves which are full of water. It has a lot more plant water content than any of the other floating species. And that is, well, maybe excluding sponge plant, but we don't have that much sponge plant out there in the delta yet for me to compare. But the 2 main species that we want to differentiate it from to get at the true water hyacinth acreage is primrose and pennywort. And both of those species have less plant water, and so what you see is that some of these water absorptions, they are much shallower in water primrose compared to water hyacinth. And again, so this is an example of pennywort, primrose, and water hyacinth, and you see that for the most part, all of the water hyacinth can be differentiated when you look at some of these fine water absorption indices. Spatial resolution. So because this imagery, and this is specific not to hyperspectral data, this is almost known as hyperspatial data, but maybe not even that. That's probably even finer. But being airborne, it can get at much finer resolutions than something like satellite imagery, which might be anywhere from 20 meters to 1 kilometer. What you see is that you get a lot of mixed pixels. You might have water in that, you might have some part of the floating mat, you might have some part of emergent, and then it becomes harder to differentiate, to call a pixel as one class. Basically, is it floating, or is it emergent, or is it water, because it's all mixed in. Whereas you look at a smaller pixel and you have mostly pure pixels. So spatial resolution matters. So because of all of these reasons, this imagery is really ideal for doing our job. I don't think this is important, but we do provide multiple, so we use a lot of techniques and we put them into a bunch of classifiers to get at the best possible classification. The results, which is probably what most of you are interested in. I'm sorry I took so long to get there. So the delta-wide acreage, this is showing our results from 2004 to 2014, and immediately the first thing you will notice is that the floating vegetation, so the FAV refers to floating aquatic vegetation, so the floating vegetation has really shot up. Compared to what it was anywhere from 2004 to 2008. Whereas the submerged vegetation is approximately the same acreage. It has been less before, but it is within that range of variability that it was from 2004 to 2008. For those of you who like numbers in hectares, in 2014 we had about 2,900 hectares of submerged vegetation and 1,550 hectares of floating vegetation. So the total invaded area in the delta has increased from 5,000 acres in 2008 to almost 12,500 acres in 2014. The cover of floating has shifted from a codominance of pennywort and water hyacinth to a codominance of water primrose and water hyacinth. So the reason I try to bring attention to water primrose is it's not less. It's about 2,000 acres of the delta today is covered by water primrose. The composition of the submerged vegetation hasn't changed as much. It is still dominated by Aegeria densa, but we'll know more this year. So just a caveat there. Water primrose is a major concern, not only due to its increasing rate of spread, especially in the Northwest Delta, but also because it is not one of the species that can be sprayed. So it's not mandated yet. So this is something we really need to pay attention to. Just a few examples of what we've seen. We've seen a huge increase in submerged vegetation over the last 11 years in Liberty Island and also in primrose. So most of that floating, the yellow that you see out there in Liberty Island is almost all primrose. And this is an example of 3 areas where primrose is really spreading up in Liberty Island. Rhode Island, basically the same story, a huge increase in floating vegetation. The entire island is covered. This shows the distribution of water hyacinth versus water primrose in Rhode Island. A couple of things. So what do we do with all of this data? We now have 11 years worth of data of where these species are. How does that help us? It helps us look at things like how much of it persists from year to year. It helps us look at things like what is the growth from year to year. So you can get at growth rates, you can get at persistence rates, you can get at the relationship between different classes of species or communities out there. So all of this data, especially when taken in a temporal fashion that is helpful, can really, really inform us and does inform us. Just a few possible reasons that we have seen such a huge increase, especially in floating out there. I went over some of these already, but part of the reason might be that the prolonged drought has led an increase in shallow habitat more conducive to their growth. And of course, there are positive feedbacks that both of them influence the environment. They are ecosystem engineers, as one of the earlier talks pointed out. Mild winters and lack of large storms. I'm not going to go into that because I don't have time. And thank you for your attention.
Okay, we have time for a question or two.
Yes?
Do they have spectral signatures for invasive weeds that aren't currently known to occur in the delta, like alligator weed and...
Could you repeat your question? Because we have it on one of the boards. We know we can hear you, but...
I can also repeat it. He's asking if we have spectral signatures of other possible invasive weeds that might not yet be widespread in the delta. Am I right? Yes. Yeah? Okay. So I'd like to... So until now, we have concentrated on the floating vegetation, the emergent vegetation, and the submerged vegetation. Of those species, we have a good idea, but alligator weed, for example, so we don't have in our database all of the species in there, and the more species you include in your classification, the more inaccurate it is possible to get at the species you're really interested in. Remote sensing is not exactly the best tool to get to rare species. I'll say it right off. It is not. It is a great help when something is really widespread, or at least starting to become widespread in the area that it is developing. Rare species, to get at rare species, unless they have something really unique about them, you know, they flower at a season that nobody else flowers in, or they have a unique signature to their flowers, or there's something unique about their phenology. Unless there's a way that you can differentiate it from everything else, it's very hard to pinpoint a rare species. So as of now, no, we deal with mostly dominants.
One more question?
Thank you.
Before everyone stands up, hang on, just a couple thoughts or announcements. For the panel discussion in the afternoon, we're putting 3x5 cards out on the table out here. So if you'd like to ask a question, we're not going to address all of them. We may be selective, but please fill out a question. There are pens and cards out there, and they'll be put in the pile. So please do that on your way out for lunch. Very briefly, everyone's on their own for lunch. If you go straight out the door here, you'll see the Memorial Union, the tall building immediately across the street. That has many lunch options. If you continue east on that same road, it becomes 3rd Street. There's a lot of options just as you walk down within, say, 5 minutes. So please be back here about a little before 1:10, and we'll see you then. Thanks very much.